Assalamu alaikum and welcome back. This is Fatimis with your grade 12 biology Telikilaas 2. In our first Telikilaas, you would remember we discussed about the process of photosynthesis and how plants are adapted to carry out this process. We talked about how plants have their many leaves which have many photosynthesizing cells that have many photosynthetic organelles called the chloroplast, and that in those chloroplasts in the thylakoid membranes you could find the photosynthetic pigments such as chlorophyll. And I told you that we'll be talking about these pigments in this class. So as I do that, my focus would be to help you understand that the chloroplast pigments can be separated by using chromatography and how you could identify these pigments by using their RF values. We'll also be looking at these two terms, the absorption spectrum and the action spectrum, and you're expected to know to differentiate between these two terms, identify which is which, demonstrating your understanding of these two terms. So let's start by talking about these photosynthetic pigments. What do you remember about chlorophyll? Well, you would remember that chlorophyll is the green pigment that makes the plants green, and you also would know that magnesium ions are required for chlorophylls to be synthesized, right? So where in the chlorophyll did you find these magnesium ions? So if you look at the structure of it, you see that in this ring part of the chlorophyll molecule, the central molecule is where you find these magnesium atoms, and this ring part is called the porphyrin ring, and that's the part of the chlorophyll molecule that's involved in the absorption of light. And you also see that it does have this quite long tail, a large part being just hydrogens and carbons to it, and this part is called the phytol tail. And this part, being hydrophobic, allows itself, this chlorophyll molecule, to embed itself into the hydrophobic parts of these proteins. So if you remember, the bilayers will have, phospholipid bilayers will have this hydrophobic region, and in those hydrophobic regions we have hydrophobic amino acids of the proteins as well. So this hydrocarbon phytol tail, being hydrophobic, allows the chlorophyll molecule to embed itself into these hydrophobic regions of the proteins, making the chlorophyll to stay in place. Now you don't really have to know to draw this structure, but you have to know where the magnesium is found and all. Now you would also remember that if magnesium ion is not present, if they are not taken up by the plants start to become yellow in color, right? You also see this yellowing if plants, just before they fall off, before the leaves fall off, so you see them becoming yellow, then orange, sometimes even red in color, which makes you wonder, do plants have those many different pigments, or is it that those different pigments are just being produced when the chlorophylls are not synthesized? Well, if you take a green leaf, separate out the pigments and have a look at it, you find that although the leaf looks green, it's not just green colors, green pigments that you have in them. You do have the green chlorophylls, but in addition to that, you find the orange carotenes, the yellow xanthophylls, and sometimes you could find this gray phyophytin, which is a breakdown component, and some of the red and purplish plants you find the anthocyanins in them as well. So although leaves appear green, it's not just a green pigment, they do have many other pigments as well. It's just that the chlorophylls, there is so much of it, you just don't see those other colors. So let's look at how we can get such a chromatogram. So that is going to be by the process of chromatography. You would remember from your O-level chemistry that chromatography is the process that is used to separate out mixtures of different pigments. So when you want to identify those pigments in a leaf, what you would do is first take those leaves and you're going to grind them up by using a mortar and a pestle, and you're also going to use a slightly hydrophobic solvent here. So the purpose of the solvent is that when those leaves are ground up, those pigments in the leaves can get dissolved in the solvent that is made, so that you have a pigmented solution formed. Also, you're going to take the chromatography paper that you want to do this on, and you're going to draw a baseline on it, and you have to make sure that you use a pencil for this. And it is important because if you had used maybe a pen that has pigments in it, those pigments can get separated out as well, and you wouldn't know whether the results that you have, those pigments that you have at the end, whether it was from the pigmented line that you drew or whether it was from the pigments in the leaf itself. So you draw a non-pigmented baseline using a pencil here. And then you're going to transfer a couple of drops of this pigmented solution that you made onto the baseline so that you have a concentrated pigmented spot on it. And then what you're going to do is you're going to transfer this, dip it into the solvent that you used to make the solution in the first place, and you have to make sure that the pigmented line that you had, the spot that you had, doesn't is not touching the base solvent here, but the paper still does. So once you do that, the solvent is going to rise up, and as the solvent rises by capillary action, the pigments that were dissolved in it, they are going to rise up as well, depending on their size and how soluble it is in the solvent itself. So the smaller molecules are going to rise up faster, the more soluble ones are going to rise up faster as well. And you can see yes, have separated out here. So before this solvent reaches the edge of the paper, you're going to take it out and you're going to mark where the solvent was at the last point. So that's the solvent front, and you're going to also identify the position where those different lines were found as well, and you're going to mark those distances from the baseline. So solvent front is marked and the position of those pigments are marked as well. And you can also tell from this particular example that because there were 4 different pigmented spots, for 4 pigments seen here, that this particular leaf that was used only had 4 different pigments in them. Okay, so if you want to find out the RF value for it, let's say how we are going to do it. So you would remember RF value is basically a ratio of how far the pigment has moved compared to the solvent itself. So you use this formula to get this RF values. For example, if you want to find out the RF value for carotene, what you would do is carotene has moved 11.5 centimeters from the baseline. So you take 11.5 divided by 12, because that's how much far the solvent has moved. The answer you would get is going to be 0.958 something. And because RF values are given to 2 decimal places, you round it off, you get 0.96. Alright. So for example, we would do for something else again. Let's say chlorophyll A here. Chlorophyll A, so that's 4.0 divided by 12, and you get the answer as 0.33. Alright. Now, this chromatogram that we have is actually slightly different from the one that I showed you earlier. So, what differences do you notice, and why is it so? So, let's see if you can answer this. For starters, you see that the initial diagram that I showed you, it has 8 different lines, which tells you that there are 8 different pigments here, whereas this one has just the 4, which tells you that this was taken from another leaf entirely, another plant entirely, because if it had been from the same leaf, from the same plant, the same pigments would be there. So it's the different plant altogether. Another thing is you also notice how those pigmented lines that you have, the positions of them are not the same as well, right? So carotene is definitely at the top in both of these cases, but you see the chlorophylls are lower in this in the one that we've given here, but slightly higher in this case, which tells you that the RF values are different, which tells you that a different solvent was used. So when you want to find out the RF values to compare, you have to use the same solvent. Alright? So let's use this information that we've learned so far to identify the pigments that I have in, say, a money plant that I have. So I'm interested in knowing how many different pigments are there in this money plant, and I also want to know what's the most abundant pigment in this. So what do? Well, I'll carry out chromatography. So I take out the leaves, make a solution out of it, use a particular solvent, separate it out, and I get the results here. And I've used the solvent hexane, acetone, and trichloromethane. And you can see I've marked the solvent front, and I've also found that there were 5 different pigments in pigmented spots here, which tells me that my money plant has 5 different pigments in it. So I could find out what these pigments are by first identifying their RF values, but I'm interested in just one of them compared to the others. As you can see, of these 5 different pigmented spots, one of them is more concentrated than the others, telling me that that's the most abundant pigment. So I want to know what this third spot is, what's this third pigment that I have. So I'll calculate the RF value for this by using those distances that it had moved, and I see that it's around 0.59. So the RF value was 0.59. But this doesn't still tell me what the pigment was. So I'll check for somebody else who's already identified what the pigment is and calculated the RF values from some other reference source. So I found this information online, and I found that this person has used the same solution, the same solvent that I have used, and he also has one pigment, chlorophyll A, that is of the same RF value. So I know that my pigment 3 that I've got is the most abundant pigment that is found in my money plant that I have. Now, I still also want to know what this pigment does though. So I know that pigments like chlorophyll A would be ones that's going to absorb light. And I know that light is part of the electromagnetic spectrum, which is this entire frequency of different waves that carry energy within them. And of this electromagnetic spectrum, light is just a small portion, the wavelengths of around 380 to about 750 nanometers. So that's the visible light. So I know that it's going to absorb wavelengths of light in these different frequencies. And I suspect that because it's green in color, it doesn't absorb those green wavelengths, because I know that if an object has a particular color, it's not absorbing those wavelengths, it's absorbing other wavelengths. So I don't think it absorbs green light, but I still want to know what other wavelengths it absorbs. So does it absorb these rest of the wavelengths still? So what would I do then? Well, I'll have to make up a solution of it, and then I'll be passing light through them of different wavelengths, and I'll be measuring how much of light passes to the other side. So I'll be measuring how much of light is being absorbed by the solution that's being made. Now you don't have to know how this is done, but you do have to know what the results tell you though. So you'd see that as this light passes to the other side, the results are going to be displayed in terms of the wavelength of light and how much of light is being absorbed by the pigment itself. So the 400 nanometer is going to be this violet blue wavelength, 700s are going to be the reds. So as I change these wavelengths of light, how much of light is being passed to the other side without being absorbed is going to be measured, and that is going to be drawn as a graph on the display that is being attached. So how much of light is being absorbed, it's going to be measured, and it's going to show a graph, and this graph that I have, that is called the absorption spectrum. So the absorption spectrum is basically a graph that shows for a particular pigment how much of light is being absorbed at the different wavelengths. So if I look at chlorophyll A I can see absorbs wavelengths of around 430 nanometers, which is going to be the blue wavelengths, and also absorbs around 670 nanometers, which is going to be in the red wavelengths. So I know that chlorophyll A absorbs these frequencies of blues and reds the most, and it doesn't absorb this green and even the yellow wavelengths, which I suspected, and that is why chlorophyll A is green in color. Now you know that it's not just chlorophyll A, you will have other pigments as well. Most plants will have a couple at least. So if I look at chlorophyll B, for example, the absorption spectrum of chlorophyll B shows me that it also absorbs some blue wavelengths and some reddish wavelengths as well, but slightly different wavelengths from that of chlorophyll A, right? So, but it still doesn't absorb the green and yellow wavelengths, so it also has a slight greenish color. Carotenoids, on the other hand, which is also the next most abundant pigment in most plants, will have an orange color, and it absorbs wavelengths of blues and a little bit of green as well, but not so much, but none of the reds and the oranges, which is why it has an orange color to them. So if you were to look at the absorption spectrum of each of all of these together, of these three main pigments together, you can still see that these green wavelengths are not really absorbed, which is why plants appear green in color. So one reason is that they have a lot of chlorophyll in them, which is the most abundant pigment, which is very green in color. But although they have many other pigments, all of them combined still doesn't absorb those green colors, which is why as they are reflected, you see the plants as green leaves, having green leaves. And you can see that each of these pigments, as you can see here, each pigment has their own peaks to them too. Okay, so the absorption spectrum, remember, is the graph that shows how much of wavelengths are being absorbed, how much of light is being absorbed by a particular pigment at each of these wavelengths. Okay, and remember, spectrum refers to the colors, absorption refers to how much is being absorbed. A similar graph is actually called the action spectrum. So from this term itself, you can tell that then having spectrum, we're talking about the wavelengths involved, and action would be the photosynthetic activity, because that's the action that's being carried out by the plants, right? So we'll first look at a short video on how this action spectrum was found, and then we'll discuss about it.
Engelmann placed a strand of the green alga Spirogyra on a microscope slide. He added water that contained oxygen-requiring bacteria. The bacteria distributed themselves randomly on the slide. Engelmann placed the slide on a microscope and illuminated it using light filtered through a prism. This breaks up light into its component wavelengths. Over time, the bacteria were attracted to areas where oxygen was being produced by photosynthesis. This was primarily where violet and red wavelengths crossed the algal strand.
You can see from Engelmann's result that photosynthesis occurs best in these blue and those red wavelengths, and the graph that we have that displays these results, it's called the action spectrum. So the action spectrum is going to be these results, the graph that shows you the rate at which photosynthesis occurs at each of these different wavelengths. Okay, so action referring to the photosynthetic activity, spectrum referring to the wavelengths of colors that are being involved in it. So this graph, it will always be in terms of something to do with photosynthesis. So it would be either the effectiveness of photosynthesis, it could be the rate of photosynthesis, or it could be in terms of the relative activity of photosynthesis. Okay, so remember the term photosynthesis has to be there, it will be there. Now looking at these two together, the absorption spectrum and the action spectrum, remember they are both graphs. One of them refers to the absorption of wavelengths, whereas the other talks about the photosynthetic activity at those wavelengths. Okay, now as you would know, it's these pigments that are being absorbing the light, and that's those that are being used for photosynthetic activity. So what could be the advantage really of having more than one pigment? Now if you had say just chlorophyll A, for example, you would notice that the action spectrum is definitely going to be limited to a small range of wavelengths, because it's only those wavelengths that are going to be absorbed for photosynthesis. But if you add to that chlorophyll B in addition to chlorophyll A, you would have a wider range of wavelengths that can now be absorbed and therefore used for photosynthesis. Add to that the carotenoids, you would have an even wider range of wavelengths available for photosynthesis. So this one advantage, this major advantage that plants have for photosynthesis, is that they will normally have more than one pigment, which ensures this wide range of wavelengths that can be used for photosynthesis to occur. So if you were to put the two together, this would be more or less how it would look like. So it's these wavelengths that are being absorbed in blue that can be used for photosynthesis, shown in black. Okay, and another thing to keep in mind is that you can see in both of these graphs is this high energy blue wavelengths that are going to be absorbed more and used more than the red wavelengths. And one advantage of this would be that that high energy can therefore then be used to synthesize the rest of the photosynthetic substances that would be produced. So what have we learned today? Well, we learned that plants have many different pigments, and those different pigments can be separated by using the process chromatography, and those different pigments can be identified by using their RF values. We've also looked at how these different pigments, their activity can be shown as an absorption spectrum, where you measure for those pigments how much of each wavelength is being measured by the pigments. You could also show the activity of the photosynthesis by the plant in an action spectrum as well, showing for each of these wavelengths how much photosynthesis occurs in them. So I hope this was useful for you. Thank you for listening. I hope you have a good day. Assalamu alaikum.